So let's see if it actually recognizes anything.
Is this same? Okay, yeah, it works. This one works.
Just can't see it either. Thank you. You guys got stuff to. Yeah, everyone. I think we're going to get started if that is okay with you. So the first couple of minutes, I'm just going to go back and review to make sure we're on the same page. I'm not sure if you can actually hear me. Is the microphone on for all of you? Can you hear me in the back? All right. So I did get a question online as to whether or not we will be continuing to stream all of the remaining courses, remaining lectures. We will. So if you are feeling ill or you're not sure if you should be coming to class or not, please stay home. It's probably better just to get well. We want to proceed with like an abundance of caution. So we will be streaming, and you don't need to worry that you're going to miss a lecture. I will also talk very briefly. From last week's lecture, a lot of you were very concerned about what material you actually needed to know. I'm going to distill it down to 2 slides. You need to know 2 slides, and that's it from last week's lecture. And you don't need to worry. The recordings are there, but I also don't want you to panic about that. And I will be posting those slides for you in the very near future so that we can all get started on studying. All right, so we're going to go back and we're going to revisit a little bit of the epidermal layers. And we're going to talk a little bit about the different layers that we were supposed to have learned last week. I'm going to just reintroduce those concepts for you today. So we are going to be talking a lot about the melanocytes. And in the previous lecture, when we were looking at these melanocytes that exist sort of in this basement layer, or this stratum basale, or the stratum germinativum, this layer was the one that we had these melanocytes that are producing this melanin, and this is sort of a cartoon version of what these melanocytes would look like. They have these long projections, and these projections are in contact with other keratinocytes, the skin cells that we often associate with skin. And we had talked about how they could release some of the melanosomes that actually contain some of the different forms of melanin, and we talked about the different types of eumelanin and pheomelanin, the etc. And we didn't really go into any more detail about that, why it's important and why melanin plays such a critical role in terms of melanogenesis. We did talk, though, very briefly, a little bit of why UV rays from the sun can actually be really damaging to your skin on multiple levels. So it is like a physical burn on your skin. We know that there's a change and there's an increase in blood flow to the area of skin that has undergone UV damage, because if you press on it, the blood flow is stopped, and then when you let it go, it comes back in. And we know that there's an increase in blood flow to those particular areas, and we know that it can have varying severities of that burn as well. And we talked a little bit, and just a heads up for you, you're going to need to go back and review these slides that we covered about 2 weeks ago in terms of the types of DNA damage, the photo byproducts of having exposure to UV that can occur to different skin cell types. And these are the most common mechanisms. So we often talk about the fact that we have these pyrimidine dimers that can form because they have an overexposure to UV. And the melanin that we're going to be talking about today is something that is going to protect the DNA in these cells from forming these photo byproducts. And so if we're taking a look, again, just as a review for you, often we have these two different pyrimidine bases, and we can either have a C or a T. And it turns out that the CC cyclobutane dimers are the ones that are more often considered to be dangerous, that might lead to carcinogenesis, that might lead to mutations that cause the different forms of skin cancer that we looked at much earlier on. And I know it seems like it's been a long time since we talked about it. It's only been about 2 weeks, so hopefully some of this will be coming back for you. All right. I wonder why this is not working. One of the things that we didn't spend a lot of time talking about is if there is a mutation that occurs within the DNA, and one of these mutations actually cause a cyclobutane CC pyrimidine dimer to form within the tumor suppressor, or the p53 gene within the DNA, this can actually be a very, very important event that will eventually lead to that cell becoming carcinogenic, dividing uncontrollably, and it can lead to the types of mutations that ultimately can cause skin cancer, depending on the cell that we find this mutation in, other mutations that might also be occurring, etc. But we often see in many of the forms of skin cancer these types of CC cyclobutane pyrimidine dimers that form in the region of the p53 tumor suppressor gene. And Dr. Sidaway mentioned that very briefly in his talk last week, and I just wanted to go back and make sure that you understand that p53 is an important gene. We want to protect it as much as we can, but sometimes we can't do so. This UV radiation is also going to affect your skin. Your skin is a reservoir for many, many different types of immune cells. That's what we're going to come back and visit again today. And will also help to suppress some of the immune responses, including the immune suppressor T cells that we mentioned just in passing. It is an important bullet point to make sure that you know. And the skin is vulnerable to having changes in the immune profile as well. So it's not just damage to DNA, it's also like affecting your immune system as well. And we also know that in addition to that, we will get the release of a number of different pro-inflammatory cytokines like interleukin-1 alpha or TNF alpha. Interleukin-10 is a really interesting molecule because it is technically anti-inflammatory, as many of you will have learned. And in some places, we think that this anti-inflammatory effect to prevent inflammation and to prevent the recruitment of immune cells to an area that's been damaged is equally important. So we're kind of rethinking the way that we look at inflammation and anti-inflammation in different ways. So if all of this is occurring, then one of the things that might happen is that we might have mutations in a particular gene where we form these cyclobutane dimers, and that as a result, we might be forming, if it's found within a melanocyte, we might start to see the formation of a malignant melanoma. We might start to see squamous cell carcinomas if it's in different layers of the keratinocytes, or we might see basal cell carcinomas as well. And we will increase the risk of developing enough of these mutations that will accumulate and accrue over time to increase the likelihood of developing these different forms of cancer. And again, the numbers not being important is just to emphasize that these are really serious. And the one thing that Dr. Sidaway mentioned at the very beginning of his lecture last week is that we need to be able to catch this early. If it's caught too late and it's begun to metastasize, there really isn't a high survival rate for melanomas, and it's still true to this day. And so early detection is something that he emphasized in his lecture, and it is really, really important. So again, just as a review, hopefully for everyone, that we're all back on the same page, that we're all kind of focusing now back on the skin. We looked at the epidermis, and again, just as a reminder, these are squamous cell epithelial cells. And the major type of cell that we find there in those 4 or 5 layers are these keratinocytes. Some of them are dying off, they become anucleated, we start shedding them, but the majority of the cell types that we find are indeed these keratinocytes. These basal keratinocytes, by the way, the ones that are at the very bottom that begin this process of forming your skin, these are the ones that are stuck to that basal lamina or that basement membrane. And again, this is that layer that separates the epidermis from the dermis. And just as a frame of reference for you, I mentioned that this was important. We have a number of stem cells that exist in the skin, and these basal keratinocytes, the ones that are at the very, very bottom of the epidermal layer, the ones that are in close contact with the basement membrane, also known as the basal lamina, these are not fully differentiated. And again, I know there was a lot that Dr. Sidaway mentioned in the last lecture, but one of the things that he mentioned was cell cycle arrest and cell cycle progression. And these are undifferentiated cells, so they're still undergoing things in the cell cycle, and they do have the potential to be able to proliferate. So these basal keratinocytes actually can proliferate if they're given the right stimulus. And so I just wanted to make that clear. But we also have other stem cells that have the same properties that are not these basal keratinocytes. And as these basal keratinocytes, they start to leave the cell cycle, they undergo cell cycle arrest, they fully differentiate, then they will form those keratinocytes in those upper layers. Now, this is why we're talking about keratinocytes. So I was hoping that Dr. Sidaway would have gone through the mechanism in a little bit more detail, but we want to make sure that you understand why these keratinocytes actually begin the process of forming melanin. So they're not the ones that actually make melanin, but you'll see why they're so important by the end of today's lecture. So imagine that you're sitting out there in the sun. So many of you have been taking advantage of the great weather of the last day or so. Did anyone sit outside? A couple of you, maybe, kind of. You wanted to, at least, right? So if you're sitting outside and you were exposing your skin to UV, and it's pretty weak UV, and you probably weren't sitting out there very long, if you were out there long enough, some of that UV light would actually affect the DNA within these keratinocytes. And some of these changes to the DNA might actually produce some of those photo byproducts, probably not to the extent where it's going to cause DNA damage that's permanent, and these keratinocytes are going to be eliminated anyways. And these photo byproducts, if you get enough of them and it's in enough of these cells, might activate some of those DNA repair enzymes that we need and are always kind of surveilling things. And especially at that basal keratinocyte layer, which is, again, really important because I'll mention this in the diagram that's coming up next. If you've been sitting out there long enough, you've been sitting in the sun, getting UV, some of the different parts of your DNA within those keratinocytes, especially in the deeper layers, as you kind of enjoy the warmth of the sunlight, they're going to start forming these photo byproducts. That means the DNA is going to be damaged, and we want to initiate DNA repair, so we would stimulate this P53 pathway. So you can understand why damaging the P53 pathway is not a good thing. or having mutations in p53. So let's assume that p53 is completely active. And one of the things that will happen is in the keratinocytes, as the p53 gene is being activated, it will actually cause the transcription program within the keratinocytes to cause the release or the formation of melanocyte stimulating hormone, or MSH. And so if you've been sitting there long enough and there's just enough damage to stimulate the DNA repair pathway, your keratinocytes are going to be releasing this MSH, and this MSH is released from the cell. And again, you'll understand why this location of where these different keratinocytes are located is important. And if it's nearby and it's not diffusing too far away, this melanocyte stimulating hormone will then bind to the M1 receptor, or the G protein coupled receptor, which is what I'm asking here. It's a GPCR, or a G protein coupled receptor, that is found on a nearby melanocyte. And we know that these melanocytes are normally found in the deepest layers on that stratum germinatibum, or that stratum basale. So the deepest layers are the ones that we're finding this. Ultimately, as I'll show you on the next diagram, through this process, now in the melanocyte, we're going to be turning on this pathway that Dr. Sidway talked about an awful lot in the last lecture. And this pathway that he was mentioning and did all of his research on is this MITF pathway, or the MITF pathway. And this is a transcriptional factor pathway. So this will turn on the transcription of different genes that are involved in producing melanin and packaging melanin, all within this melanocyte. So again, before I go on, because I'm going to tell you in advance, I'm going to rewrite this out again for you, along with the team that puts together the study guide for you. But it's important that you know the different steps. Do you have any questions or things that I didn't really clarify for you, or things that made it a little bit more confusing? And again, show you this in a different form on the next slide. But is there something on this slide that isn't clear right now? So there's a lot on this particular slide here, and not all of the things that are on this slide are ones that you necessarily need to know. So what we've been talking about is these deep layer basal keratinocytes. So this is in the bottommost layer. You'd have to imagine that because this is where we find the melanocyte, different colored cell, melanocyte down here, keratinocyte, keratinocyte, keratinocyte over here. This is deep, so this is a basal keratinocyte. And one of the things that happens is you've been sitting out there, you're enjoying the UV, and it is able to penetrate the first 4 to 5 layers of your skin, all the way down to this layer down here. And one of the things that UV will do is it will induce those photo byproducts, those cyclobutane, CC, and TT pyrimidine dimers that we just reviewed at the very beginning of today's lecture. And as a result of these different photo byproducts accumulating within this basal keratinocyte over here on the left, one of the things that will happen is the p53 repair pathway, which is what you are seeing here, the p53 is going to be upregulated. It is a repair pathway that normally detects any damage to your DNA. And one of the things that happens is as p53 is increased, it will actually cause the increased production or activation of a synthetic enzyme known as POMC. And this is that POMC. And POMC is the element or the genetic element that will eventually cause the formation and release of that melanocyte stimulating hormone. So this again is a slightly expanded version of what I mentioned in that previous slide. So this keratinocyte over here is releasing, in response to UV damage, it's releasing melanocyte stimulating hormone, and it gets released from this cell. I'm hoping that we're all kind of on the same page so far. It's not the only thing that happens, and for the sake of clarity, we're not going to go through all of these different factors. The only one that I'm also going to be mentioning is as UV damage occurs here, we just went through this melanocyte stimulating hormone pathway in response to UV damage. That UV damage also will cause the initiation and release of not just POMC to create MSH, but it will also cause the release of SCF, which is stem cell factor. And stem cell factor is going to be important on the next cell as well. Okay, so these two factors being released from this keratinocyte on the very lowest basal layer of the skin in close proximity to this neighboring melanocyte. So melanin production actually begins in keratinocytes, and it actually goes back to keratinocytes. It doesn't necessarily begin in the melanocytes directly. The other factor that I want you to make sure that you know gets released from keratinocytes is this SCF or stem cell factor. Are we all okay so far? Is there anything that I need to go back and repeat for you? No? You don't need to worry about any of these other genes that get turned on through the P53 dependent and independent pathways. The ones that I'm going to look at are MSH as well as the stem cell factor. All right, so let's assume that all of that's happened, and now in this melanocyte that's nearby, on that basal lamina, on that basement membrane, we have this nearby melanocyte which is now detecting some of those products that are being released from this nearby keratinocyte. So it's detecting the MSH that's getting released, and it's also able to pick up the release of stem cell factor as well. So again, it's really small. I hope it's bigger on your screens, but MSH will interact with the melanocyte or M1 receptor found on the melanocyte. It will then turn on a cell signaling pathway that includes like increased cyclic AMP, increasing protein kinase A, and this turns on and activates cyclic response element B, which is known as CREB. Why all that is important? Because MSH is active on the cell surface, it's going to activate transcriptional programs within the melanocyte to cause the increased production of MIT-F, which is shown up here. And in fact, if you look at the other factor that gets released from these basal keratinocytes, which is that stem cell factor, on the right, that stem cell factor, which is shown as sort of that little green ball, also being released by the keratinocytes after exposure to UV. So this factor over here, it's going to also work on the same melanocyte, and it's going to work on a different receptor system, and this is not a G protein coupled receptor. This dimerizes, and this is known as the c-KIT receptor. And the c-KIT receptor activates MAP kinase to phosphorylate and stabilize this MIT-F that is being produced. Now, why is this MIT-F so important? And again, you don't need to worry about all of the details here, but if you look down here at the bottom, these other pathways will eventually cause the pathway that's shown on the right, which is pigment synthesis. So this MIT-F turns on melanin production, where it also packages the melanin through structural elements that get turned on into those melanosomes. And that means that this melanocyte now, in response to both MSH as well as to the stem cell factor, is now upregulating the production of MIT-F, and MIT-F in turn is upregulating the production of the melanin and packaging it into those melanosomes so that it can be released to the nearby keratinocytes. So keratinocytes cause increased melanin, and that increased melanin is used by these nearby keratinocytes as well, because they're not capable of actually producing these pigments. And we think that this is important because melanin acts sort of as an umbrella to shade the nucleus, which is what is being shown here on the diagram on the right. So as more and more pigment gets picked up, as more and more melanin is being formed, because we have MIT-F transcriptional pathways activated, that means that the nearby keratinocytes, which are picking up all of that melanin being produced, will actually now be shaded or have their nuclei shaded and protected from any further DNA damage. And that's why your skin changes color as you start tanning or as you start getting exposed to more and more UV light. Now, I'm also just going to mention, because time is like really, really short, and you're going to have to correct me if I'm wrong, your final exam is like really close to the end of the semester, right? It's not the end. It's like April the 9th or something. And you guys finish on the 3rd, so it's like 6 days after. So I'm going to tell you in advance because I need you to start thinking about it. I know you have other things going on. There are going to be several questions related to this mid-half pathway. There are going to be several questions related to the molecules that I described here. And we are going to rewrite this pathway out for you so that it's easier to understand or read through. But this is a pathway that you need to know for your final exam. So one of your short answer questions, like one whole question, will be devoted to like a problem scenario, like what might happen if you have a mutation in the M1 receptor and c-KIT receptors that don't allow binding to occur in the presence of like UV, what downstream pathways would be affected, what might happen to the keratinocytes. So problem-based, application-based, but you need to understand this. So again, this c-KIT receptor also found on the cell surface of these melanocytes. It's working through that other factor that gets released from the nearby keratinocytes that are UV exposed. All of these lead to the increased activation and production of MIT-F, and this then in turn will upregulate other genes that are associated with different types of melanin. And again, we talked about that in that last lecture before we had Dr. Sidaway in. And this melanin is then transported and released from these melanocytes and taken up by other nearby keratinocytes. That whole pathway and this diagram, not that I'm going to ask you to draw this diagram, but understanding like the different elements of it, you don't need to memorize the details of different transcriptional pathways that get affected here on the middle panel, nor do you have to memorize all of these other genes that we didn't talk about, like ETI and basic FGF, etc. But the fact that we have stem cell factor and MSH that gets released after P53 is activated is an important one. If you look very carefully, there's also this other interesting sort of gene over here as well, and this XP gene is actually also really important. It's also a gatekeeper. So not only do we have P53, we also have this P53 alternate pathway that also involves other gene products that is called XP. And this is also really key because if you don't have XP and you don't have this functional repair system in your DNA, this can actually lead to cancer in these individuals. So this is like very brief exposure. So some of you sat outside in the sun, like fairly weak sun, so it's still not full summer yet, so the sun isn't directly overhead, nor was it really warm out. But you can imagine just going out into the sun for like 2 or 3 minutes. If you have a mutation in the XP pathway, this is sort of the result of what happens. You get like skin damage incredibly quickly. And that's because you don't have this XP gene repair pathway. Your DNA in your keratinocytes and your melanocytes are all being affected. And unfortunately, if you don't have this, your survival rate is actually very low. And this xeroderma pigmentosa, this genetic autosomal recessive disease, is actually a very serious one. It often results in different forms of skin cancer. And so all of these checks and balances on DNA repair and skin are incredibly important. We take them for granted, and we know how important it is because if you have a mutation in this, it is a very low, like 9-year median survival rate, means that young children are not going to be able to survive much past the age of 9 years. Often they have melanomas and different severity of melanomas. And so these DNA repair checkpoints are incredibly important. Not that you need to worry about anything on here. I'm just pointing out that this is sort of an addendum. You don't need to worry about it for your final exam. It's just to highlight the importance of the fact that these DNA repair pathways and shading and protecting the nucleus and having all of this in balance is incredibly important. We take our skin for granted, but we shouldn't, because we know that very severe skin disorders can result if we are missing some of these repair pathways. Okay, so I'm going to transition a little bit, and we're still talking about skin, and we're going to bring up sort of the last module and a half, which is the microbiome on the skin and in our guts. And in addition to that, I also wanted to introduce that concept about immunity and skin immunity on this as well. This is also going to be covered on your upcoming final exam, so please make sure that you are comfortable with the material, that you understand the model system. We're going to start it today, we're going to finish it next week, and then come back next week also to talk about skin and the development of organoids or mini brains to help us understand situations like schizophrenia and how we can use your skin to grow mini brains to help us understand schizophrenia, and then we'll come back to the microbiome in health as well as in disease. And I thought we'd go back and revisit a little bit about skin and what might happen on your skin. So your skin is covered with bacteria, a little bit less nowadays because everyone's kind of washing their hands a lot more frequently. But just to put things in perspective, so skin actually has some innate immune properties that are associated with it. And I mentioned at the very outset of the module that we're covering now on skin that we need intact skin. So skin is actually a barrier to prevent microbiological agents from entering into our bloodstream, and we have all of these layers of protection between the surface and to the blood vessels underneath. But one of the things that happens is often we break our skin. So you might have, for example, a little injury. So it might be in the form of a trauma, it might be a little burn, it might be a little cut. And for most individuals, those basal keratinocytes, if you cut your skin, because they're proliferative, they will actually migrate up the edges of the wound, and then they'll start dividing, and they'll fill in that little cut. And you get complete resolution or complete healing. But we also have this other situation that we need to think a little bit about because it's going to be a big part of the model in this particular paper that we're going to be using. You might know of individuals that actually it takes them a long time, like if they get a cut on their skin, it takes them a long time before their skin can actually heal itself. And while it's healing, we call this sort of an ulceration. So it's partially healed, but it's not completely resolved. You don't get full coverage and full replacement of that intact epidermal layer. And again, sometimes it resolves and it actually takes a little bit longer, but it might actually go to form like scar tissue. And you can also go the other way, like scar tissue, if you continually damage that same area of skin, that chronic scar can actually be broken down and you can actually form an ulcer as well. The problem becomes when you have an unresolved or you have not completely healed this damaged area, this initial injury persists, the skin doesn't repair itself, this ulceration is lasting, and then if you continually damage that area of the skin or it's not healing fast enough and it's continually being exposed to different microbes or other factors, this can actually lead to what is known as Marjolin's ulceration or Marjolin's ulcer. And this is a form, it may not seem like it because it's like drastically discolored. This is actually a form of squamous cell carcinoma. And this is actually the model system that we're going to be thinking about or looking at in that paper that I showed you earlier. So again, our skin has a lot of really unique properties that are associated with it. Unfortunately, we weren't able to kind of get you out into the clinic because of things that were kind of happening around the hospitals and not wanting to take in too many students because of the coronavirus. But one of the things that if you were to go, and you can still go, by the way, we're hoping the coronavirus thing gets resolved pretty quickly, and I'd love you to go and visit some of the different clinical sites. One of the things that happens a lot is what is known as a surgical site infection. So, as the name kind of suggests, surgical site infection, you go to the hospital, you go in for surgery, and then while you're in the hospital, you get an infection. Can you think of a reason why that might happen? So you go to the hospital, is that a pretty sterile, safe place? Okay, you go to the OR, you know where everyone's scrubbing, everyone's wearing masks, got the caps, gowns, everything. Sterile, 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 sterile. Do you expect to get an infection there? No. But if you go in for a surgery, about 1 to 2% of the population, even in Toronto, will get a surgical site infection. And then you have to think why. Can you think of a reason why? Yes? Okay, so as it suggests, the immune system goes down. Why? Why does your immune system go down if you get surgery? Any ideas? What body temperature are most of you? It's not a trick question. Honestly, it's not a trick question. So around 37, so somewhere between 36.5 and 37 degrees. What happens when you go in for surgery and you know it's going to hurt if they cut your skin? What do? They give you an anesthetic. And when you're given an anesthetic and you're completely under, your brain shuts down. And as your brain shuts down, because you're going under and you're not quite sleeping and it doesn't really look like sleep, but one of the things that happens is your body temperature, because it's controlled by your brain, the hypothalamus also starts to slow down. And your hypothalamus can't regulate your body temperature. And so one of the things that happens is your skin gets colder. And that again, up until about 5 years ago, was very common practice. You go in, you get an anesthetic, you're put under, your skin grows colder, and as your skin grows colder, your immune system, your immune functions in your skin actually depress as well. So when you cut your skin, even if it's completely sterile, and you go back and recover outside of the OR, you're going to be re-exposed to different bacteria. All of your immune cells, because your skin has been cold, or it used to be the case, actually those immune cells in your skin are no longer quite as active, and it means that for that brief period of time, you are going to be exposed to bacteria on your skin that your immune system just can't handle, because there is no immune system at that point in time. So people started to realize there's a lot of immunity going on in your skin, and that actually began with the literature on surgical site infections. We know that immunosuppressants, if you are on immunosuppressants because you've had some kind of transplant, that these immunosuppressants that are suppressing your immune system, even though you've had nothing to do with the skin, suppressing your immunity actually increases the rate of skin cancers. Suggesting again, if you're suppressing the immune system and you're getting more skin cancer, and you're not affecting DNA directly, but you're affecting the immune system, that also suggests that your skin has immune function associated with it as well. We also know you have a ton of bacteria on your skin, and normally that bacteria doesn't cause you many problems, except when you have like an immune compromised situation. So all of these things are issues that people have known about for a while, but there's been really no one great study to kind of link all of these different factors together. So what is this link between wounding the skin, and is there a link between wounding the skin and inflammation, which might or might not draw in immune cells? So again, we talked about now like why interleukin 1 and interleukin 10, so pro-inflammatory and anti-inflammatory. And then if it goes far enough, and we know that if you have like an unresolved, damaged piece of skin where it's not healing itself and it's continually being damaged, it causes an ulceration, which leads to Marjolin's ulcer, which is a form of squamous skin cell cancer. Is there a link between all of those things? And this is one of those papers that tries to address and link all of these things together. So that Marjolin's ulcer that I showed you earlier is a very aggressive form of squamous cell carcinoma, and it can take place in areas of the skin that have been damaged before. So you get an ulcer, damage it again, you get that same patch of skin, not completely healing, and then at some point your skin just gives up, or your immune system in your skin gives up, or both happen, and then you get squamous cell carcinoma of the form that is called Marjolin's ulceration. It's through damage to your skin. We also know that your skin can react in a number of different ways. So one of these is known as keloid scarring. And some of you might have seen individuals that have this. So keloid scarring I showed you earlier. So you cut your skin, and usually you get a little remnant where your skin was damaged, right? You get scarred. So most of you have scars, whether it's psychological scars from being there at UT or physical scars or whatever. But most of you will have had a scar. But some of you will see individuals that actually have a very different type of scar, which is called a keloid scar. And in some ways, it kind of looks a little bit like a tumor, because not only does the skin grow and cover that cut in your skin, but it actually forms a bump. So it literally forms an outline on that cut in your skin, and it's actually a bump, because it's a benign tumor. The skin has overgrown, in other words. Have you ever seen one of those? Like literally, like I've cut it here, and instead of just healing normally and leaving a little physical mark, the scar actually grows out and is actually a bump on your skin that exactly matches that cut on your skin. And this is a form of keloid scarring, and this is a form of benign tumor formation as well. So again, we have a lot to kind of think about in terms of the role that your skin plays and repair and inflammation and other things. And these benign tumors that often grow, these keloid scars, have also sort of led to a number of different research lines that look at skin wounding as well as cancer. And again, we're trying to make this link. Is there something that happens during the process of skin wounding that leads to Margolins ulceration? What is that link? What model systems can we look at? And one of these model systems that has been developed because people started with keloid scars, like benign tumors, so they normally don't cause any problems. They look terrible because no one wants a scar that's like really pronounced on your face, for example, or on your hands. And people get plastic surgery to remove that keloid scar. But is there a different model that we can use to examine how skin grows malignantly, or how we get the formation of cancer, not a benign tumor growth, but a true cancerous one that is related back to wounding? So instead of looking at a benign tumor, let's look for a different model system. And one of these is known as RDEB, so recessive dystrophic bullosa of the skin. And this is where you have a skin blistering disease, and you often have the same cycles like Marjolins ulceration. So you have a scar, it doesn't completely heal, and then you damage that area of the skin again. And you might be thinking, who would do that, right? Like, who would purposely go back and damage their skin again on the same place? Well, think about like if you're walking, and you need to be able to walk, and it's on the bottom of your feet, and you're going to consistently walk, and it never really heals itself, and then you're going to go back and walk again, where you're using your hands a lot, and it's going to be damaged. And so this inherited disorder, this RDEB, actually has a lot of the properties that is very similar. So we get this squamous cell carcinoma. Almost 100% of individuals that have this autosomal disorder is going to have at least one form of squamous cell carcinoma in their lifetime. And that means that, again, they're at risk of developing this form of skin cancer. And the key question is, what causes this transition from having like damaged skin and an ulceration that doesn't really heal? And is there a link between not fully healing and the immune system? And then if it doesn't heal, then it actually forms a cancer, like a squamous cell carcinoma. It's not, as Dr. Sidaway mentioned last week, it's not lethal, but it's also not completely benign either. It can cause other issues. So what is this link, and why do these patients that have like problems with their skin not healing, leading to cancer, and then also having some other issues, like what are all of these links that we can use and sort of discover? So again, in a little bit more detail, this disorder, and this is, again, there are a number of different classifications of it. It depends on what layer of the skin is being affected, and we can call it different things. So it could include like the epidermis itself. It could actually affect the dermal layers, or it could even affect like the basement membrane. So all of these are susceptible to being like chronically damaged, not completely healing, etc. And so there are these three different types depending on the area. So the epidermal type, the dermal type, basement membrane type, and these are the full names of them. And the one that we're going to model in this particular paper involves more of sort of the dermal layers being damaged, and this is the one that the paper is going to be addressing. I just wanted you to sort of see why we are calling this RDEB. This is an individual that actually has RDEB. You can see their hands sort of have these incompletely healed ulcerations on their knuckles at the top, on the side of the satellite. On the left side for you, and you can see that they have had probably a number of incidents where there's been damage to their skin over time. This hand also has had the same types of damage associated with it. So this is where we're going. We're going to try to unwrap this paper a little bit, try to understand the context of this paper a little bit, and to understand why this is important. Use that idea of the skin bacteria, how we analyze that, and then talk about that as a jumping off point into the microbiome in sort of the next module. So why don't we take a 10-minute break now? It's a great place to take a break. We'll come back and we'll begin unwrapping this paper a little bit. Sometimes I'm not sure how much of that we have to go back and kind of paste back together. All right, folks, we still have a couple of minutes before we have to get going, but I had a quick question. So, as you know, your term pass or your final exam is actually really close to when classes end. Are there any specific things that you would like to have as a class for your final exam preparation? Just so I can get started on it a little bit earlier, is there anything that you can think of off the top of your head? We will be doing exactly the same thing that we did for term test 1. So if you show up to your last tutorial, there will be like a practice set of questions similar to the final exam. You'll take it up in your tutorial. You can take that with you. So that's like extra practice questions is on our radar. One suggestion was to have like online quizzes so that you can practice some more of the multiple choice. Do you have any other suggestions? Do you guys want to do well on the final exam? Don't care? Okay. All good to me. Yes. Yeah, Miss Sita? Yeah, I'm thinking that I'm hoping it gives you enough time. I don't know what your exam schedules are, to be honest. So I'm hoping that we'll do something similar for April the 3rd. We are registered for Exam Jam. And then that Exam Jam session will also be streamed if one of the streaming team shows up. We will like provide some kind of audio and video for it regardless. But in that Exam Jam, I will be kind of creating sort of a distilled version of all the most important slides. So that is sort of on the radar as well. Any other suggestions? No? Yes. Yep, okay. Yep, so one of the suggestions was to just have like a final exam specific discussion board thread, and I'm happy to do that. It's a great suggestion. All right, so if you happen to think of anything off the top of your head later on, please email me. I'd just like to get as early a start as possible. We want to make the exam as well prepared for you as possible, and I can only do that if I have time to prepare. All right, so I'm going to hope that this streams normally, hopefully. And unfortunately, the other mic died, so I can't actually walk back and forth, which is not my favorite. All right, so if we're looking at wounded human skin, or if you have psoriasis, so you have like skin that kind of really flakes off and it kind of is red and it's always kind of like looking like it's wounded. It's just a condition that people have. One of the things that seems to happen is that in a wounded skin, or if we have like an excess of like psoriatic skin, so very unusual, it's not a form of cancer, but it is something where the skin is being removed much more frequently than it should. We also see that there is an upregulation and an inappropriate expression of a molecule known as beta-1 integrin, which many of you will have heard of like as an extracellular matrix molecule sort of binding protein. And one of the things that this means is, especially in wounded skin, as it's trying to repair and as it is also found in psoriatic skin, if we're trying to model a disease with wounding, we need to understand the basic process. So the upregulation of beta-1 in wounded skin, incredibly important. An upregulation in psoriasis, which is again dead flaky skin, and an excess of dead flaky skin, also important. And one of the things that is well known about the beta-1 integrin pathway in keratinocytes is that if we start overexpressing in the keratinocytes too much of this beta-1 integrin, we also will get an upregulation in that skin. So damaged, wounded skin or psoriatic skin, we'll also get an upregulation of the extracellular signal-related MAP kinase, MAP kinase, which is known as MEK1. Okay, so damaged skin or psoriatic skin, we get beta-1 integrin. As a result of that, we also get an upregulation of MEK1. I'm hoping those are kind of linked for you and it makes sense for you. So one of the things that we try to do, and we talked about this way back at the beginning of the course, is to come up with animal models of these different types of disorders. And if we were to take and we were somehow able to overexpress just the activated intracellular kinase, MEK1, if we were to somehow turn on MEK1 all the time, so it's like having too much beta-1 integrin, too much MEK1 activity, could we actually reproduce either psoriatic skin, like excess dead flaky skin, or like wounded skin that's trying to repair itself and it may overgrow itself and may form something that looks like a keloid scar? If we could somehow do that would we have a good model that we could use in animals to model wounded skin, inflammation, and subsequently cancer or overgrowth? And so this is sort of the end result of all of this. So if we take MEC1 and we were somehow able to take the constitutively active, which means you don't need beta-1 integrin to be turned on to turn on MEC1, but we somehow turn it on constitutively, could we then see some kind of skin disorder? And what we do is we can constitutively turn on MEC1, and we can put it under the control of an involucrin promoter, and we call this mouse that has overactive or constitutively active, it's always turned on MEC1 or MAP kinase 1. Under the involucrin promoter, we call this an INV mouse. And this EE will make sense in a moment, but the IN or INV part comes from the involucrin that I'm going to talk about in a moment. And when we correctly overexpress only in the skin this activated MEC1, which is normally associated with psoriatic skin or with like wound healing, this transgenic mouse actually has chronic skin inflammation, so it has psoriasis, and it also has epidermal hyperproliferation. And in fact, it starts to grow these benign tumors that we will call a papilloma. So it has these nodules on its skin. The skin just like overgrows itself when we express this MEC1 in normal skin cells. Now, the next slide is an important one. This mouse model is an important one. Again, just to get you ready for the final exam, which is only 4 weeks off, so you can start worrying now. So worry. It involves like knowing this model and knowing and being able to describe how it could be used. So I'm going to show you what this model looks like. So this is MEC1. This is the MEC1, and the reason why it's called an INV or INVEE is because we create point mutations in the MAP kinase that change the serine into E, so that's one of the E's, and we have two of those, so we actually have EE. So that's the INV part, which is over here. And this is all under the control of the involucrin promoter. And why we use this is that we only want this MEC kinase to be expressed in skin cells. We only want it in keratinocytes. We don't want it in melanocytes. We don't want it anywhere else. We want it in keratinocytes. And these keratinocytes, if we overexpress MEC1, it should look like it has psoriasis, it has hyperproliferation of the keratinocytes, etc. And so this involucrin promoter is really important. And the next slide explains where this idea is all coming from. So normally, we find involucrin as a product of these keratinocytes. So it's normally produced by keratinocytes. So it makes sense that we are taking the promoter region of the gene that's produced by keratinocytes and using it as the driver for this. So one of the other molecules that is formed by neighboring keratinocytes or epidermal keratinocytes is loricrin. And loricrin will bind to different gene elements, and it will produce in the keratinocytes, it will produce this molecule known as involucrin. And so this means that because loricrin turns on and activates the gene for the involucrin, which is a separate, different molecule, and we're not going to go into what this molecule normally does in the skin. This means that this molecule is a marker for skin cells, okay? And we have the promoter, we have identified the genetic region, which means that any gene that follows after the involucrin promoter is going to be turned on specifically in skin cells. And so this promoter is like really important, and that's where the INV part comes in. So it's a marker for terminally differentiated keratinocytes, and we're using this promoter that is only found in terminally differentiated keratinocytes. No other cell type has this. Melanocytes don't have this. Hepatocytes, no other cell type, just terminally differentiated keratinocytes have this involucrin molecule, and therefore it has a gene, and this gene undergoes transcriptional regulation by lorocrine. So if we just have the promoter, we can drive gene expression in a specific manner. And this is why we put MEC1 under the control of this involrucrin promoter. And if we look at the transgenic mice, and you have to look fairly closely, you'll actually see like extra nodules of skin. So it forms these like large bumps, like there's hyper proliferation, too many layers of skin are being formed, and it forms these nodules that are benign tumors directly on the skin, because the skin is just the keratinocytes are way too active, they're hyper proliferating all the time. And we can take histology and we can see that it is indeed many extra layers of keratinocytes, again, they're benign at this stage, but we want to use this as a model system for potentially things that look like squamous cell carcinomas, like in RDAP and in other molecules, and in other systems. So again, do you have any questions on this before we go on? I'm going to tell you that you need to know the in-B mouse model. You need to be able to show that you understand why MEC1 is important, link it back to the beta-1 integrin, link that back to like when you have psoriasis or when you have like excess skin that's flaking off, or when you have wound healing, we normally overexpress beta-1. We normally therefore also overexpress MEC1. Why we're using the involrucrin promoter system. It's only found in terminally differentiated keratinocytes. So if you don't understand any of that, please come and see me during office hours. I'm also going to be posting some practice questions of the type that you'll get on the final exam, both short answer as well as multiple choice, so you can start practicing that. When would be a good time to start posting some online questions for you? Would next week be all right, or is that too early or too late? Too early? Next week sometime? Okay, so I'll give you some examples of multiple choice related to understanding this system. So a couple of other things before we go on. And sadly, if you looked at the news and you are in immunology, or if you've taken like M250 or M350, or if you're taking a course now with Dr. Jasti Singh, you might have heard of Fiona Watt. And Fiona Watt is on this particular paper. She passed away just a few months ago, and she is the immunologist that actually started examining these really interesting gamma delta T immune cells. So Fiona Watt's group, or Professor Fiona Watt's group, was actually the first to identify that there might be a set of immune cells that are actually pro-tumorigenic. Again, that didn't make a lot of sense. How can immune cells actually promote the formation of tumors? And what does that look like? And what are these cell types? Because they are not classical T cells, what do these cell types actually look like? And she had actually demonstrated that there are some macrophages, so we know our skin has lots of macrophages, as well as peripheral gamma delta T cells that might actually promote the formation of tumors. It was heretical at the time that she first actually suggested that we have this other class of T cells that actually promote the formation of tumors. But although there's controversy, many people have actually found and identified these specific subclass of T cells within the immune system. So we know they exist. Whether they do the things that this paper and others suggest, still debatable, but I'm going to present it sort of in the way that they presented it. So this gets into going back to our original question. So now that we have looked at the in-B mouse model and the rationale for developing the in-B mouse model, overgrowth of skin, which looks a lot like the overgrowth of skin that we see in different forms of like RDAB that might result in like squamous cell carcinoma. We know that there might be immune cells that might be important in the progression of this, but we haven't examined it yet. We know that these mice, when they overexpress in keratinocytes, the MEC1 that is commonly associated with skin disorders, they do form these benign tumors that we call papillomas. But can we make all of these links between inflammation, papillomas, and some of these different immune cells that will cause the skin cancer? And what is this link with the bacteria on our skin and the immune cell and the formation of cancer? So all of these questions that we started off with, we're going to start using this NV mouse model, and I'll start taking you through some of the data today, and I'll finish off the story next week over these next few slides. You are never required to know like the size effect or to be able to draw any of these things. This is just to go through some of the data with you. So if we look at the mouse model, if we look at, for example, on the back of the skin, which is usually the place where most of these experiments are done, it's also to prevent the mouse from kind of biting or trying to clean off its wound, so it can't really reach the back of its parts of its back really easily. So on the back of the skin of both wild type and the NV mouse, you start measuring like these papillomas at this particular wound site. And again, for all of these different models, there's usually a very linear response. So if you make a wound on the back of the NV mouse, the larger the wound size is, the larger the papilloma that results. Okay, so not unexpected. We have like, you wound the skin, normally tries to repair itself. I talked about sometimes you get overgrowth. And in this mouse model, the larger the wound size is, the larger the papilloma size is going to be. So you get overgrowth, and it's linear. So the bigger the wound, the bigger the papilloma. If you start looking at, do immune cells start to come into the wound in both the NV as well as in the wild type, and we look at total immune cell infiltrate, and we're just going to use a general sort of marker for immune cells. And for those of you who take immunology, you'll know many, many other cell surface markers, but we'll just use CD45 sort of as a general marker for immune cells migrating to this area. And if we were to take a look, we get immune cells moving toward the area of damage. Not unexpected, right? But one of the interesting things is they were significantly more abundant in the NV skin both before wounding occurred. So even before wounding happens, we get a lot of immune cells. We don't know what type they are. We just have a lot of immune cells in the layer of the skin, and we get more of them after we get wounded. So again, remember, in the NV mouse, you wound the skin, it doesn't heal, it actually forms a benign tumor. So one of the possibilities, or one of the things that this suggests, is that because we have immune cells in the skin, and because we get an overgrowth of skin, one of the classic signals that lead to immune cells migrating to an area of damage is some kind of cytokine or inflammatory marker. So this suggests that inflammation is somehow playing a role in this whole process, and this is sort of one of the things that they want to be able to examine. So is there a correlation? So at rest, the NV mouse has a lot more immune cells happening, and after damage or after wounding, wherever you're looking at, every single time point that you're looking at, we always get an increase in the NV mice, which are in these white bars, versus like the wild type mice. So at every stage, before wounding, after wounding, anytime that you're looking at this, we always get more immune cell infiltrates measured by CD45. Is there a correlation with inflammatory markers? Is there like inflammatory markers also like elevated in this NV mouse? And one of the things that they look at in the skin of these NV mice, and this is in relation to wild type. So this is in comparison to or relative to wild type. So any bar that you see above 0 says that it's higher than you would have in wild type. So we see all of the different types of things that we would normally expect. So we have following inflammation, we'll have activation of NF-kappa B or nuclear factor kappa B. It's often associated with inflammation, and it's also, again, trying to make that link associated with cancer as well. And of the many, many different genes that were examined in the NV epidermis relative to the wild type, the authors were able to see that there are many genes that are normally associated with the inflammatory pathway that were elevated in the NV mouse versus the wild type. So again, suggesting that in this mouse where we have like something going on with the skin and it is forming these benign tumors, and then after you damage the skin, you get these really large papillomas. They already have like a baseline activation of the inflammatory system, and you don't have to memorize any of these different markers. The fact that I'm highlighting NF-kappa B will be important later. But if we take a look, we have a number of these different factors. And in fact, it's not just in the skin. If you look systemically, so within the bloodstream of these animals, we see all of these other factors like thymic stromal lymphoprotein, poetin, TSLP, tumor necrosis factor alpha, interleukin 6, all of these are elevated systemically. So these mice, and all we've done is we put in a constitutively active MEK inside skin cells of these mice, and they form tumors. These mice are chronically inflamed. They're always having an inflamed status, and we already know that we have, before wounding, we have a lot of this immune infiltrate. So, and again, it gets even higher if we do wounding and we form these tumors. So something is going on. There is a link between activating the skin, causing it to undergo this proliferation, having inflammation, and then potentially developing these tumors. And we're also getting a lot of CD45 immune cells in the skin already, but especially after activation. So again, trying to make this link with all of these different things. So here's this really interesting experiment, right? So this interesting experiment is something like this. So again, we're kind of looking at the incidence or the formation of these papillomas, these benign tumors, and we are looking at these in V mice. So in V mice, we get a lot of tumors, not unexpectedly. I told you about the MEK1 promoter driving like proliferation in the terminally differentiated keratinocytes. And then these authors do this really cool thing. So they've just shown in that previous figure that one of the things that is elevated in the bloodstream, in the skin tissue directly, is TNF-alpha. So do you remember that from the previous slide? Are you still there? Are you not there? Okay. If there's anyone online, you can hopefully you can follow along too. So they do this really interesting thing. What happens if we somehow alter that immune response? So I can't really easily take away that TNF-alpha, but let's try something a little bit different. So they didn't generate, but they have access to these other knockout mice. And these knockout mice are deficient in the receptors for TNF. So any cell type that is part of the immune system, when they use these TNF receptor 1 and TNF receptor 2 knockout mice, so these are the two major receptors that normally bind to TNF-alpha, which is elevated systemically, elevated in the skin, part of that inflammatory cascade, they shouldn't be able to respond to TNF-alpha. Right? No receptor for TNF-alpha, therefore you can't respond to TNF-alpha, right? Are you with me? Are you kind of with me? I'll take kind of. Kind of? All right. So mice that are deficient in both of these receptors, they exist. You can get mice that have like both TNF-alpha receptor 1 and 2 knocked out, meaning that they can't respond at all to TNF-alpha. And then we have this involucrin constitutively active, forming these papillomas. You wound the skin on their back, they grow these massive tumors on their back, and that's being shown here. And in the previous slide, we tried to establish that, okay, there's something weird because one, there's already a lot of TNF-alpha, there's a lot of inflammation, and we get immune cells that are already in the skin. Is there a link between all of this? So the authors are trying to address that issue. So they knock out these receptors, and they want to know, so if this tumor that is forming, do you actually need to have your immune cells coming to that wound? And is that the reason why you get this papilloma. So they knock out these receptors for TNF alpha. TNF alpha is still high, and they cross it with the involrukin 1 mouse. So the involrukin 1 mouse has now, in this scenario here on the bottom, this mouse has MEC1 being overexpressed, so it's hyperplastic in the keratinocytes. And we cross that now with this mouse line, where these mice, the offspring of this cross, so the offspring of the involrukin crossed with the TNF alpha receptor knockouts, have like involrukin MEC1 overexpression. Keratinocytes are still able to proliferate, still have that really high TNF alpha response, but any of the leukocytes that are being generated from the bone marrow, they have no receptors for TNF alpha. They can't detect that there's high levels of TNF alpha. So this is this other new line that is being produced. And this again is really cool, but also really important that you understand this. So they've just crossed this, they've just added or taken away the ability for any of the immune cells to respond to TNF alpha because the receptors are completely gone in these mice. They wound the back of these mice now, which is what happens. And then they examine what is the incidence of the papilloma. And the papilloma response is dramatically reduced. So you can see down here at the bottom, the papilloma incidence in these mice that have INV, just like these mice at the top. They still overproduce the cytokine, like TNF alpha, but they don't have the receptors for TNF alpha, and so they don't migrate to that area, and they don't also form these papillomas. So do you all see that? Do you need some time to think about that? Are you amazed by the power of molecular biology? It's pretty cool. It's a very interesting experiment. But they were like, again, showing that somehow there is a link between tumor formation, the resident inflammatory status, inflammatory signals, and the immune cell all happening within the skin. So suggesting that maybe some forms of skin cancer, especially like the RDEB, although we don't know how that all works, might involve these similar mechanisms. So sorry, I know it's a really heavy lecture and you need time to think a little bit about that. So these TNF receptor ablation experiments where the immune cells, so leukocytes in particular, don't have any of the receptors, can't respond to TNF, don't go to the skin, don't really respond to in the same way. They don't form these large tumors after wound formation. And so what they did was some immunocytochemistry on the wounds as well as on the papillomas. And one of the things that they started to see was at the edges of these were these gamma delta T cells. And again, really, really interesting because there wasn't a lot known about this about 3 years ago about these particular gamma delta T cells. And they also saw that there was a reduction of the CD4 positive. So we started off with general CD45 positive cells, but CD4 positive T cells were also selectively reduced in this double knockout mouse model where there's no TNF receptors at all. And again, it suggests that something is going on with the immune system, something is going on where there's inflammation around dividing cells in the skin, and this is what we're going to come back and pick up on. And I'm going to mention that all of this might begin, and why you get skin cancer might actually begin because you have bacteria growing on your skin all the time. It's part of your skin microbiome. We talked about the skin microbiome and acne, and we kind of use that sort of as a way to look at skin. But when we look at some of the different things that we'll go back and look at next week, if we look at all the things that eventually lead to NF-kappa B activation, the one thing that you look at here is that there are a number of bacterially related molecules, and the one that we're going to look at in particular are the ones that involve flagellin, so this toll-like receptor 5 that you see here, and we're going to take a look at that as potentially a way in which skin cell cancer might develop outside of like having UV damage, etc., like how this might happen in certain types of genetic disorders. Just one last note, we are going to end a little bit early today, so I'm hoping that's okay with all of you. It's a very busy time for everyone, and I know there's already a lot of material. There's still more to come, so we still have like 3.5 lectures to go. I want you to start thinking about the final exam. I don't know what your schedules are like in the last 2 weeks, and tell you that this is the earliest it's ever been for HMB 322, so you're going to literally be studying right up until the last minute. So I'm just finalizing the final exam now. Do you want more multiple choice or less multiple choice? 15 or 20? 20. Okay. I have no idea. All right. 15 and a half. Okay, I like that. All right. Okay, so for those of you who are saying 20, is it just because you feel more comfortable with like having 20 and you're used to multiple choice? Okay, fair enough. Honest answer. Could we meet in the middle? Could we have like 17? Are you good with 17? Maybe? Okay. All right, I'll take 17. So you are going to have these one-line answers, and so you have to go back and think. Like in that last term test, like I didn't do so well on the one-line answers, or I didn't give like specific enough responses, and then these 4 short answers. So again, one of them I can already tell you involves like the NB mouse. The NB mouse model is going to play like for one whole question. Both parts are going to involve the NB mouse. So if you are completely lost at this point, and in the beginning of the next lecture, please come and see me sooner rather than later. I'm happy to help you. Like I won't look down on you at all. Like maybe I'm just talking too fast or talking too slow. It's more important that you do well on the final, so please let me help you. But one whole question is going to involve like the NB mouse model. So if you're lost or you couldn't follow along what I was kind of going through on the lecture slides today, see me earlier rather than later. You only have 2 hours. Generally in the past, it's been more than enough time for people to finish as well as to review their work before submitting it. Again, you have to think back to what happened in the last term test. I think most of you are okay with time. You just get like 2 extra short answer questions. So like, will you have time to be able to review over the course of 2 hours? I hope so. We do have a final exam preparation. If you can't make it down on that day, that's fine. We'll try to stream it. We'll try to make sure that everyone has access to it. I will create sort of a distilled slide deck for everyone and post the slide deck just like for term test 1. If you have other ideas for getting you ready for the final exam, please let me know. The sooner you let me know, the more likely I'll be able to do that. I want you to have the best possible final exam experience that you can. It's important for me too, because I hate seeing students kind of who know the material but are just like really stressed out and can't really get their answers down because they're stressed out. So we want to make sure that you're well prepared. All right, so for those of you who are presenting this week, good luck. I'm sure you'll do a fantastic job. Again, have a great week. Be safe. Make sure that you're in communication with your TAs. I'll see you all back here next Tuesday.
I don't think it's cut out. The connections for like people watching see the exact thing. You can ask the questions in the chat.
The immunology will be interesting because there's a lot of controversy around the gamma delta T cells. And Fiona Watt's work, I like it, but a lot of people don't like it. I want to ask you, so looking at 3.5 months of I like the building entire Genetic antibodies. Yeah, maybe. I'm going to try to convince you that all of this is actually related to skin bacteria too. So why does that